Good morning, Biology 102. Today I want to walk you through 6 pieces of evidence for evolution. I have them listed on this first slide and they will go in order. So we're going to start with fossil record. So fossils are imprints or remains of organisms that lived in the past. They can be amberized, they can be imprints, they can be mineralized like petrified woods and things like that. But they are some kind of remains that we can then look at and study. Scientists typically will then put fossils in a ordered sequence as they appear in rock layers, and basically to tell a story of time old to time present. This ordered sequence of fossils is known as the fossil record. And again, both of these things are going to help us tell a story about how our populations are changing over time, and that is at the heart what evolution is. It's a story about how these populations are changing over time, from time beginning all the way to time present. Now, fossils, some of the ways that we sort fossils into this fossil record, we use things like law of superposition. The deeper the fossil, the older it is, the more newer fossils are on top. So the position of the rock layer sometimes can tell us about the age of a fossil, or at least order the sequence of fossils from like sort of older to newer sort of organisms. We also radiometrically date different fossils. So we use radioactive isotopes, the ratios of the mother isotopes and the daughter isotopes, in order to figure out how old something is, to give an actual age to a fossil. And so this gives us the ideas of like how old the Earth is and then when different living things have existed. Index fossils are fossils that fossilize really well. Think of things like clams and different snails and things like that that have hardened shells and parts that like stick around in rock layers really well. Index fossils typically will cover large areas, fossilize really well, and so they're often, we know that they lived in a particular time, and so we can compare different rock layers in different areas, right? And then determine when those rock layers existed based on these index fossils. And transition fossils, just like over here, these transitional fossils show almost like baby steps, right? Or transitions between an earlier population to what we see at present. And it gives us kind of missing links, again, to help us tell a story of how populations are changing over time. Over here, this whale, it's showing some transitional fossils in the flipper, right? And how the femur, the tibia, fibula, tarsals, metatarsals, those particular positioning and the bone structures changed as you went from a tetrapod walking on all fours on land to something that is aquatic with a flipper that is moving water. So it's a little bit of a different structure. But we have these transitions that allow us to see how that population changed, moving from land back into the water over time. Another piece of evidence for evolution is biogeography. This is the geographic distribution of species across the planet. We can use the movement of plate tectonics, so when plates have been together and then moved apart. Using indexed fossils, we can date when those plates were together or when they were apart, and like kind of across the oceans, look at different like sort of species that have occurred together. At the heart of biogeography is this principle that species present in an area today are there because they evolved from ancestors that inhabited those regions before. This tells us, using plate tectonics, fossils, and this principle explains why we have so many marsupials in Australia. This plate came away from another plate and isolated a marsupial ancestor that has then led into all of these different marsupial groups that we see in Australia. That's a little bit different than what we see in like other places like Africa and other places around the world. Another piece of evidence for evolution is comparative anatomy. This is comparisons of morphological body structures between different species. Homologous structures are those structures that share a common ancestry, like the forelimb over here. All of these mammals share a common ancestor, the rhapsid mammal, who had a similar sequence and collection of bones in the forelimb, right? The humerus, the radius and ulna. So the radius and ulna are like kind of the radius follows your thumb and the ulna follows your pinky bone coming down in your forelimb. There are analogous structures that are similar because of convergent evolution or environmental factors, but do not share a common ancestry. Usually these look similar maybe at first glance, but upon closer inspection, these structures are quite a bit different. A good example of this is wings. So bat wings up here, you can see are finger wings, and they have finger bones inside of them. Bird wings are a whole four-limb sort of wing, so they're using all of these larger bones. Insect wings don't use bones at all. They use like kind of an exoskeleton, sugar chitin, in order to build their wings, and they're very different. You can imagine dragonfly and beetle wings. Those have developed over time in a very different way structurally. Vestigial structures are what we call leftover structures. So these are structures that maybe an ancestor used for some purpose. Remnants, or the whole structure remains today, but they are no longer used. So these structures are things like, my favorite is in snakes, there are a few species that have vestigial legs inside. You don't see it from the outside, but if you were just to dissect out the snake, you can see those little vestigial legs that links them back to lizard like ancestors, right, that had legs. And so they no longer use these structures, but there's remnants of those structures that still remain. Another piece of evidence is comparative embryology. This is comparisons of early developmental stages between different groups of organisms. The reason why these are highly sort of conserved is because there are genes that run these early developmental steps and stages, and these collections of genes have changed from one group of organisms over time throughout the different groups. And so plants have their own sets of developmental genes, fungus, animals, and even within animals and plants, you have like different collections of them. And so this can also help us tell a story of how populations have changed over time. Molecular biology is using DNA and protein sequences to predict most recent common ancestors, or how different groups of organisms are related to one another. At the core heart of molecular biology, the core principle is if two species have genes or DNA sequences that match closely, biologists are going to conclude that these species share a relatively recent common ancestor. If we look over here at this table, you can see humans are often said to be closely related to or share a common ancestor with chimpanzees. And we can look at the DNA. You can see here that we share a little over 98% of the same DNA with chimps. If we go down to gorillas share 97-ish and a little bit of change to chimpanzees. So we can see that humans share a little bit more of a match sort of DNA sequence with chimps than like sort of gorillas and chimps do as far as this particular chart. And we can use this information then to predict and sort of sort organisms. We often will build phylogenies or evolutionary trees to show the relatedness and how these like changes of molecular DNA protein sequences have changed, and then the relatedness of these particular groups over time. The last piece of evidence is evolution in action, right? Evolution that we see today. Lots of people think that evolution takes too long and that we can't see it in our lifetimes, but there are some instances where evolution moves quite quickly, and we can see evidence of it in our own lifespans, or in even a few short years. Examples of this are things like antibiotic resistance, bacteria changing and adapting over time so that they can resist the impacts of antibiotics. Over here, I show a plate with antibiotics on it, and you can see things like penicillin no longer can kill or inhibit those sort of bacteria. Bacteria have resistances and ways around those particular antibiotics, and this is sort of accumulated in our bacterial populations over time. Pesticide resistance and insecticide resistance is the same. There are applications of different insecticides like DDT that worked really well at the beginning, but a few insects, mosquitoes, whatnot, could survive it. And those based on some genes that allow them to like either pump the pesticide away from them or keep that sort of toxin away from their cells can actually live. And those genes allowed them to live, pass more of their genes onto the, sort of future populations or the next generation, and cause these resistances and these pesticides to build up over time, so that some pesticides and insecticides no longer work today. Lots of people think that it's only in small stuff like bacteria that we can see it, but we can also see even in large organisms like lizards, we can see horn length changes. Even within a decade, we can see, or a number of years, we can see how horn length size has changed due to predation pressures. So this particular lizard is hunted by a predatory bird, and over time, this top horn length has gotten larger and larger, and you can imagine that helps protect it, right? So the ones with the larger horns get to survive and pass more of their genes on to the future generation. So hopefully this gives you some ideas of some pieces of evidence for evolution. I will see you next time.